Hi everyone, this is Himanshu and this is the beginning of the biotechnology under the science and technology. So, in the biotechnology, let's start with understanding the basic terms. That is a DNA. When we say a DNA, it is a molecule that all the living organisms carry in every cell in their body. DNA is actually a code that holds the detailed instruction for the building of an organism. So it's a code. So in humans, this information is laid out in the long linear strands of the DNA in the nucleus. So they have the long linear strands, and the spelling of the linear is wrong. It is linear, linear strands of DNA in nucleus. And so, rather than being one long DNA strand, we have a DNA in the form of many smaller and more manageable pieces, and which we call as the chromosomes. So the chromosomes in human, actually there are 3 billion pairs of the chromosomes, and they are divided into the copies of 23 chromosomes, one from the mother and one from our father. So with a 6 billion base pair in every cell. The chromosomes actually influence everything from a person's hair color to a susceptibility of a disease. So for example, the chromosome 1 actually contains the gene that controls whether you are susceptible to the forms of the malaria or not, or the chromosome 16 has a gene that influences the hair color. So that is an example. Some people have the missing chromosome or the extra chromosome, or an inverted chromosome, or what we call as a mutations, and these mutations causes many disorders. So these are generally what we know as a chromosomes. And when we talk about the DNAs, the DNA base pairs exist. DNA bases are actually connected with a hydrogen. So that when we say the types of the DNA itself, there is an adenine, which we call A, and thymine, which is T. So generally what happens, an adenine and thymine, so they always form a one pair, it always connect to each other. Similarly, guanine, that is the G, and cytosine, that is C, they always form the pair in the DNAs. So adenine always pairs with thymine, and the guanine always pair with the cytosine. So this is the basic properties of the DNA itself. Now the next one that we're gonna discuss is the DNA profiling. So let us write it heading here, DNA profiling. Now, the DNA profiling, this is also called as the DNA fingerprinting. It is actually a creation of the biometric database comprising the DNA information of an individual. So DNA information of an individual. So this is what the DNA profiling means. Now when we say that we are establishing a DNA profile, so generally what happens in every individual, 99.9% of the DNA, there is a similarity between all the individuals, human organisms. There is only 0.1% variations, and this 0.1 which actually differs what we call as a VNTR. That is actually the variable number of tandem repeats. Now these are actually unique to the individual DNA, and this actually constitute the DNA profile of that individual. So this is actually what defines that how a particular organisms how it behaves and what would be the characteristics, how it makes it different from the other organisms. So the DNA profile of an individual varies in some regions in terms of the number of times a sequence of the nucleotide base pair is repeated. So we can say that counting the number of times these base pair sequences are repeated in these variable regions in DNA profile of an individual is established. And when we say the application, so if we have these DNA information or DNA fingerprinting, it can be used to identify of person, means identification of the person can be easily carried out if we have all this information. Similarly, it helps in determining the biological relationship to establish actually the parentage or the viability of the organ transplantation, etc. So this is what it is generally. So when we say genes here, so we need to understand one more thing. Genes. Now, every cell in an organism contains all the information needed to manufacture every protein in the body. So I repeat that every cell in an organism contains all the information which is needed to manufacture every protein in this body. And actually the genes is a strand of the DNA which actually act as a storehouse of that information. So it's more like an instruction book, So the genes are actually carries for the particular trait is what we call as a genotype, and the physical manifestation of the instruction is basically what we call as a phenotype. Now, so we can clearly say is that if the gene exists, that is a sequence of bases in the section of DNA, they can easily affect the phenotype, because what it exactly is basically, the physical manifestations. So how does a gene affect the phenotype? Is through the two things. The first one is what we call as a transcription, and other one what we call as the translation. Now, transcription, in which a copy of the gene's base sequence is made, that's a transcription. And the translocation is in which that the copy is used to direct the production of the protein. So we can say that basically the transcription is regarding the gene's base sequence, and the translation is regarding the protein of a person or a production, that how it should be. So this is basically about the genes itself. So we can say that in transcription, actually the gene's base sequence or the code is copied into the middleman molecule, and that is called the mRNA. That is mRNA. This is actually act as a middleman, which what it does, that it maintains the gene's base sequence, and when it made, then it copies all this information to the one that is called the mRNA. So this is what it is. The next thing that we're going to talk about is the genome sequencing. Now, the full set of the DNA present in an individual organism, it's called its genome. So let me write it here, that it is a full set of DNA in an individual organism is called the genome. Is called genome. So basically, when we the mapping out a person's entire genetic code, and this whole process is what we call as a genome sequencing. So in a person's genetic code, there are actually 3 billion base pairs, and if we sequence all those 3 billion base pairs, then that's called genome sequencing. So by creating a genome sequence, we will be able to test the specific genes to detect the presence of the mutation associated with the genetic disorders. And within the next few years, the experts expect the turnaround time to improve and the cost to drop so much that the analyzing a person's genome will be no more expensive and zeroing in just one gene, and that's actually pretty much good. There is also a concept relating to this. This is what we call as a next generation sequencing. Now the next generation sequencing allows it, it's a sequencing technique actually which allows to decode the multiple DNA fragments at the same time in an automated process. So when we say the next gen sequencing, it means that everything will be automated and it can decode the multiple DNA fragments at the same time. So this is mainly about these things. So in the next section, we will learn about the gene therapy and the mitochondrial DNA and the nuclear DNA. So let's jump to the next chapter. Thank you.